So next up, we have Dr. Feldgarden. So Dr. Feldgarden is a staff scientist at NCBI and the National Institutes of Health in the US. So these are the group that run that NDARO database that Dr. Sang talked about. I try and remember to use titles, but sometimes I forget. But and talked about and kind of covered in kind of details on the high level. So what Dr. Felgarden is going to talk about is specifically the AMR like gene prediction tools. So how do we take those genomic sequences and actually predict the presence or absence of AMR genes or mutations related to AMR, etc. Their expertise, they have a lot of expertise in this area, as Dr. Sang kind of intimated, in that they're one of the main developers and the kind of project lead for the AMRFinderPlus tool. This is one of the kind of main AMR gene detection tools associated with those kind of three big databases. That'd be the ResFinder tools, CARD and the RGI tool, and then NCBI and the AMRFinderPlus tool. But then they've spent a lot of time working on the problems of AMR genomics, and I think even some metagenomics in a wide variety of different organisms, and dealing with a lot of the pain and suffering that different organisms can bring to that problem. If you're ready to go, Mike, take it away.
Let me just share my screen and get set up.
Yeah, we're a little bit ahead of schedule, but that's never a bad thing.
Okay. Before I start, Finn, is my title slide visible?
Yep. It's nice and clear.
All right. First, just before I get started, I'd like to thank the organizers for giving me an opportunity to talk about some of our work, as well as just sort of give a brief overview. I think this is a really, I started in genomics back in the dark ages when genome centers were doing Sanger sequencing. So it's really been sort of a fun ride to see how things are going. And I think in terms of using it for AMR, thinking about antimicrobial resistance, I think there's been a lot of progress over the last 15 years. So I guess to start with, the real point, one thing to think about when you're thinking about, well, what tool do I use? And of course, the problem is when you're a developer of one of these tools, you're always tempted to say, well, you should use my tool. But actually, the tools you should use, they should align with your goals. There are really there are sort of applied uses such as surveillance. These often focus on good genes. So genes where we feel really confident, if you see that gene, you're probably going to get the resistance phenotype. They're known to have an effect. Another applied use is clinical work. And what a computational scientist might refer to as an edge case, oh, that's a really weird error that was made. That's a problem for a clinician. So these applied uses are often lead one to be more conservative. On the other hand, there's research, and this involves gene discovery, and there you might want to cast a wider, less precise net. You might want to look at things like efflux pumps that are found within a species, common in a species, because you want to say, well, maybe there's a novel mechanism. So you really, to a certain extent, need to understand the goals of the tools that you are, or the tools that you are using, why they were put together. This was talked about in the first talk. I'll just sort of revisit it. This was a slide I took from the REACT group. But there are multiple mechanisms for antibiotic resistance. Antibiotics can be pumped out of the cells. Changes can happen to make it hard to get antibiotics into the cell. Targets of antibiotics within the cells can be altered. There can be alternative enzymes. So yes, the antibiotic works in stopping some kind of process from going to A to B, but there's another way the cell has to do it. And there's also inactivating enzymes. So the antibiotic is somehow modified in such a way that it can't kill the cell. And in terms of thinking about, well, how do we find these things in genomes? Two of the primary mechanisms that most tools look at are point mutations and or small insertions and deletions. Those typically involve target alterations, efflux pumps, and decreased uptake. And then there are also acquired genes, and these are often, and by acquired, I'm using that somewhat loosely. This is often something that's sort of variable within a species. So it's not, let's say, a mutation in gyrase, which every organism will have. It's something that was acquired, let's say, through horizontal gene transfer of some sort. And these can involve efflux pumps, but they also involve these alternative enzymes, as well as inactivating enzymes, and sometimes decreased uptake. A third mechanism is gene disruption, and that can be promoter disruption or the gene or an actual functional gene. Most tools to date don't really have a good handle on that. They really focus on these first two mechanisms. Another thing to think about when you're picking a tool is what kind of data do you have and how much work do you want to do up front? As was mentioned in the previous talk, a lot of these tools start from assemblies. And one thing to think about, of course, is when you do that, what you are how the tool works will depend on the quality of the assembly, as well as, in some cases, the annotation tool. If it's identifying genes in the genome, this can affect results. Obviously, if your assembly doesn't assemble the gene, if your assembler doesn't assemble the gene, then you won't be able to detect it regardless of what assembly-based method you use. And another thing to think about is if you're using draft assemblies, so these are often Illumina only assemblies, you can sometimes, if you have two very similar genes, the draft assembly process will actually squash the two of those closely related genes and only assemble one version as opposed to both. So let's say a strain has a KPC2 carbapenemase and a KPC3 carbapenemase. Those can differ by one nucleotide, and sometimes assemblers will only assemble one version. There are also read-based methods, and some of the concerns to have here is that mediocre seek read data, and so most people can identify, okay, this run just didn't work, we're not going to submit it, we're not going to use it. And likewise, high quality data is always easy to assess, but if you have a lot of data where there's some bad runs, this can really you can really get into some problems when you're using read-based methods, especially with allelic variations, because it's hard to know, well, is that do I have two versions of that gene, or is this just sequencing error? With these methods, you also need to understand how the reads are processed, how they're mapped to references, and so on. So to a certain extent, it's a black box method. And depending on the tool, often these methods might lack positional information. So if you want to know, well, okay, I found a SOL1 gene, but what's it next to? You may not get that information from some read-based methods alone. Another thing to think about are nucleotide databases versus amino acid databases. So the amino acid in most cases describes function, leaving aside things like promoter regions and whatnot. The advantage of nucleotide analyses is they can be faster. And you can also search large read sets with those kinds of analyses. But sometimes they can be inaccurate at fine scale because the proteins and nucleotides don't always correspond. In terms of the closest protein hit may not be the closest nucleotide hit. And many of these tools are hybrids, so they may look for point mutations as well as they then also look at protein sequence. So that's another thing to think about when you're looking at tools. The key things are, again, so how do we detect these methods? So there are really sort of three basic methods, and the first is BLAST. There are actually some other BLAST-like tools, but I'm from NCBI, so I'm probably obligated to just talk about BLAST. This is basically just looking for sequence similarity, and if you've ever used NCBI's graphical user interface, if you found a sequence and you want to BLAST it, that's what you have. That's the tool that's used. It's easy to understand how it works because typically you can get the BLAST output from multiple tools and use that to say, okay, well, why did I see this? What's happening there? So it's easy to reconstruct what went wrong. And this can be used with nucleotide-based methods or protein-based methods. Kmers, this is talking about what Kmers are and how to use them could be an entire seminar themselves, but basically what you're looking for This is typically used with reads, although they can be used with assemblies. You're basically chopping your data up into small sequences and then using often 25 bases long, 41 bases long, and using that to screen either assemblies or read sets. They can be very fast. And one advantage is that they can search large read sets such as microbiome data. So if you did an entire NextSeq run, you can search that very quickly with Kmers. And this isn't an AMR example, but NCBI, for instance, near the start of the COVID pandemic, actually used a Kmer-like process to search various read sets that had just been submitted to the short read archive, not assemblies, just the reads, to look for read data sets that had SARS-CoV-2 in them. So it can be, they can be mechanism agnostic to a certain extent, particularly if you're using, if these are tied into prediction. So a lot of sort of the machine learning tools that are used to predict phenotype will use these. And they're often also tied into, like I said, because they're mechanism agnostic, they're often tied into phenotype prediction. A third approach are known as hidden Markov models. And again, that could be a whole talk in and of itself, but these are basically alignments of where you take alignments of known proteins. So I take, for instance, all the KPC family carbapentamases, I build an alignment, and then using a hidden Markov model, that essentially identifies conserved domains of structure and function, again, based on a multiple sequence alignment. They typically use protein sequence for speed and computational reasons, but you can use there are some related tools that use nucleotide. And one thing is that they are based on biological structure, not arbitrary identity thresholds. So there is manual curation involved, but they are looking, so for instance, in class C beta lactamase is looking for the omega, they often will ping off on the omega loop, that kind of thing. So it's not just saying, well, if it's 90% identical according to BLAST, I'll say it's this protein. There's actually some biology directly involved. The other thing to think about is, do these tools use manually curated cutoffs, or is there what I call one rule to bind them all? So for instance, I'll talk about this in a bit, but ResFinder uses just one identity threshold. Other tools may say for this particular gene, we're going to use a higher or lower threshold. That has some advantages and disadvantages. It's obviously a lot easier to figure out, well, there's just one rule. Sometimes you have to go figure out what the rule was used if each gene has a slightly different rule. Another thing to think about is what you need in your output. So what do you need reported back to you? So for instance, a lot of tools give you the closest hit versus the best estimate. So just to walk through an example, if you found, let's say, a sequence that is 99% identical to the KPC2 carbapenemase, that is not actually the KPC2 carbapenemase. And if what you need is basically to go into some output and grab this gene symbol, this KPC2, a certain tool may not deliver what you need. Just to walk through the example, KPC2 is a carbapenemase. KPC33 is an inhibitor-resistant cephalosporinase, but it differs by one nucleotide change. And KPC8 is an inhibitor-resistant carbapenemase, and it differs by two nucleotide changes from KPC2. And I can tell you from looking at at MicroBiggie and some other tools we have at NCBI, there are a lot of unknown, uncharacterized KPC proteins. So you wouldn't want to call one of these, given the variability within this family, even though they're all closely related, you might not want to say, report the gene symbol of KPC2. You might want to just report KPC. Also, point mutation detection. Most of the large tools now do detect point mutations and acquired genes, but that is something to think about. The other thing to think about is what I call broken gene detection. And I don't think any of the tools have done a really great job with this. And so it's easy to detect something that's near full length or full length. But when you start having frame shifts you don't want to necessarily, or you might at least want to know, well, yes, I had a 99% identity hit, but there was a frame shift or there was a stop codon in there. That can happen sometimes with certain tools. You want to be able to know, tools also may or may not report whether a gene is partial and whether or not they have frame shifts. And this is really important for things like the porin-based mechanisms, something like Klebsiella. Also, depending on what you need, you may want descriptions of genes. And by that, I mean, is there a phenotype? Do you get a gene product name? That kind of thing. As well as, are there online tools that are available that you can hook into? I think Kara's talk was really nice in describing some of the additional things you can get from, let's say, CARD. We at NCBI, we have a reference gene catalog that if you see, well, what is this gene symbol? We have several tools that allow you to go figure out, well, this is what it might actually be doing, and give you more information, links to literature, that kind of thing. And this is just the sort of, I mean, the previous talk went into this in much more detail, but things to think about when you're also thinking about a tool. If somebody created the tool and they haven't updated it in 10 years, that is not going to be the best tool. I can tell you from our own work, we get different results, even just using versions that are a year or two old. So that's important. Also, have there been descriptions of how genes are included? So there are some very important curatorial things to think about, like, are full-length genes included? Are only full-length genes included? What kind of curation is going on? Are there have the curators thought about things like, well, some of these genes might be different lengths. Are we sure that the start sites are correct? And this can actually can affect things like identifying the best hit. And then there's also things like, are leader peptides included? Are certain other things removed, such as these sites found in integrons? Again, how are gene symbols reported? I touched on that on the last slide, and this is something where I think that the next talk will really, I think, both show how much variability there is in reporting gene symbols, as well as ways to sort of get a handle on how to compare results from different tools. Are there links to the literature in the database, and are possible phenotypes reported? And the reality is, often you don't know this until you start using the tool. I saw this in the Q&A, and so I'm glad I put this slide in. There's sort of a very large caveat that is worth mentioning, and that is there have been a lot of studies now, or an increasing number of studies, where people look at the correlation between genotype and phenotype. And in just sort of perusing the literature, I can't go through and summarize that there are many, many studies now. But it really looks like for some organisms, the genotype, is actually giving you a really good indication of what the resistance phenotype is. We did a study in 2019 looking at 3 foodborne pathogens, Campylobacter, Salmonella, and E. coli, and this was over 6,000 isolates. We found over 98% consistency, and a more recent analysis done by our partners at the National Antimicrobial Resistance Monitoring System, using sort of this was using a version of AMRFinderPlus that was 3 years old, suggests that we're well above 99%. The problem is, for other organisms, there are problems. And so, for instance, there's this excellent study I note here, where they looked at Pseudomonas aeruginosa, and this was a yeoman's work. They really did a lot of work. They not only used sort of traditional, looking for genes and point mutations, they used machine learning approaches. They even did gene expression. And for some drugs in Pseudomonas aeruginosa, they still didn't get, to the level of where you're at, 98, 99% consistency. And so this is a problem. I think Acinetobacter is another difficult organism. And as was pointed out in the previous talk, a lot of the confounding issues can be gene expression. This matters in some organisms, for some drugs, sometimes. And the current tools really don't get at this very well, other than perhaps looking for point mutations in promoter regions. And another thing, of course, is when you have a drug that's just come to market, there may not be a lot of work done on what the resistance mechanisms are. So I think it's sort of a mixed picture. I briefly just want to talk about sort of some of the basic differences. When I try to figure out how our tool is performing and are we making mistakes, I end up looking at a lot of papers. And so the three tools I've seen that are really used commonly are ResFinder, RGI, which is the card tool, and AMRFinderPlus. With ResFinder, it can use assembly or read data. It's a nucleotide blast-based method. There is a single identity and single length threshold, although there can be some, you can change those. It has the advantage of being very fast. I think this was in older versions with older databases, this was a problem, but I think it's pretty much disappeared. But the one thing to keep in mind is when you're using nucleotide databases, the closest amino acid hit is not always the closest nucleotide hit. But I think given the depth of their database, they've pretty much solved that for a lot of genes. And they also have an online graphical user interface, so you can just upload your data. You don't have to use the command line. RGI, which is by the card group, we heard a lot about that, so I'll just be very quick. One of the key features I like is that there's sort of there are indications of how strong the evidence is for phenotypes. I think that's a really useful thing. It can accept different kinds of sequence. It's blast-based, but it has manual cutoffs. There's an online graphical user interface, and I will say the ontology they have. So if you can look up some of the ontological terms that come out that are found in the output, that's a really nice tool. As somebody who curates our own database, it's very useful. AMRFinderPlus, I'll talk about it in more detail. Just as an example, I needed to pick one, and in the interest of time, I picked the one that I know best. It uses a protein database. So even if you use nucleotide sequence, we're doing a translated blast. It will accept nucleotide sequence or protein sequence, but the database is based on proteins. It uses a hybrid approach of BLAST and HMMs. The cutoffs are manually curated. There is explicit partial and internal stop identification within genes, so we'll report that has happened. There's no online GUI, but we do have data for over 700,000 isolates available in MicroBiggie. And at the end, I'll show you, there are some links that if you want to go look at it yourself. But these are, it is a command line only tool. So I just want to spend the last part of the talk just talking about using our tool as an example of some of the things I've talked about, why you use it, what we're trying to accomplish, our goals, and so on. So we developed, just for context, I'm in the pathogen detection group at NCBI. We have a multi-agency collaboration, not just within the US, but we also get data from Public Health England, as well as hospitals, as well as data from GenBank. And our major goal is we try to take all of these genomes and put isolates into context for both food safety as well as clinical outbreaks. But obviously, once you have genome data, you can also look for AMR data. But the key thing to note from here is our scope is large. We typically get hundreds or thousands of isolates a day that we need to process. And then our users are often surveillance systems, so they need something simple. They need output that is relatively simple. The large scale we're talking about, hundreds or thousands of genomes a day, requires concise information. So the kind of output we provide can't really be artisanal. You're not going to look through multiple columns, rows, and tables. A surveillance system simply needs to know, am I seeing X number of genes? So we need a concise, discrete signifier that conveys appropriate information about the genotype and hopefully the phenotype. And that signifier is the gene symbol. And I walked through this example earlier, but again, the point is if a surveillance system is looking and we say KPC2, we have to be certain that we are saying it's KPC2. We obviously don't want to misidentify it, and we don't want to say that something that looks very similar but we've never seen before is in fact KPC2. So we really need to nail that down. That's sort of our goal in a lot of this. We also use it partly as a curation tool. And so that's another thing where we use it for. But AMR Finder Plus, we use a curated database. As I said, we use HMMs and BLAST. So essentially, we take protein data from annotations. We take nucleotide sequence. And I should point out, it doesn't have to be a genome. We use this if somebody submits an allele and sequence and says what beta-lactamase allele this is, we just run it on that one sequence. But for genomes, we use protein and nucleotide, and then we subject it to both HMM analysis and BLAST. We currently have in our isolates browser, we have data on over 952 pathogen, 952,000 isolates. That's actually out of date. I made this slide 2 days ago, and it's now up to 953 and a half. That's the kind of scale we're working on. The reason we call it AMR Finder Plus is because we've expanded by request from our agency partners to look at virulence factors as well as different stress response genes, so acid, biocide, metal resistance, and heat resistant genes. These are, and they're partly interested in, well, if I'm using a biocide in my meatpacking plant, am I also seeing selection for resistance genes? This is an optional feature. Our database has almost 6,000 resistance proteins, and we have close to 1,000 resistance mutations in 15 taxa at this point. As was talked about earlier, I won't go through this in great detail because the first talk did such a nice job of laying things out. But we get our data from a variety of sources. We've collaborated with domain experts. We have monthly or every couple of month exchanges with CARD. We also are the curators of note for beta-lactamase alleles, quinolone resistance alleles, and mobile colistin resistance alleles. So obviously that's a source of data. We have used or incorporated for the HMMs information from ResFAMs, TigerFAMs, and we also construct what we call NCBI FAMs. And then we do a lot of validation to make sure that our HMMs are actually hitting the things that are in our database so that they work. Just one other thing that, because I touched on this earlier, to get into how we assign this gene symbol. AMR Finder Plus, we have a hierarchical structure. So obviously if something's 100% identical to BLAST, by BLAST, we know what it is. Let's say the KPC2 carbapenemase. And we know what it's going to be. KPC2 has what it does. It's been characterized in the literature. We know it's going to be resistant to, confer resistance usually to carbapenems and other beta-lactam antibiotics. But we may find something that's a little more divergent, and we can say it's in the KPC family. And this is identified by the HMM. It's likely resistant to carbapenems and other beta-lactam antibiotics, but there may be some variability in that. And then if we find a more distant protein that doesn't really seem to match anything, we just might call it, we may only be able to say based on a larger scale HMM where we take all the class A beta-lactamases and look at them, that, well, it's a class A beta-lactamase. And the key thing is we have cutoffs. So if something is really divergent, we will say it's not a beta-lactamase. And that's actually an important thing for tools to do. This is, you know, again, with manual curation, regardless of what method you're using, this allows to have some biological intuition in your software. Yeah, so this is just to summarize what I said before. One thing about HMMs, the utility of HMMs, I just hidden Markov models. This was just sort of a gee whiz thing we did a couple of years ago. We actually took GenBank protein sequences that had the name beta-lactamase in them and weren't things like cloning vectors or synthetic constructs or things like that. And we found that only 11% of the sequences out of 1 million appear to be beta-lactamases, and only 20% of unique proteins, that is a unique protein sequence, appeared to be beta-lactamases. We've tested this more, but the one test we did is people have gone through and looked for putative metallo-beta-lactamases, for instance, from metagenomic data that have been functionally characterized. AMR Finder, one study looked at 21 of these. They had 3 that they had thought were metallo-beta-lactamases. Functionally, they weren't. So they had 18 that were and 3 that were non-functional, but seemed like they were. And we nailed it with our HMMs. There's a more, and that's described, the data are from this study. There was a recent study that used 3 tools, ResFinder, RGI, and AMRFinderPlus, to look at beta-lactamases from viral genomes, which is a really interesting thing. They had about over 11,000 putative beta-lactamases, and these 3 tools basically converged on saying that only about 50 or so of those 11,000, 50, that's not me misspeaking, were actually beta-lactamases. So there, you do have to think about, is the tool precise enough, particularly if you need to be conservative for your utility. Just one more thing, and then I just want to show a sort of an example of output and talk a bit about sort of what I call the good and bad. Our optimal use in our pipeline is to use nucleotide, protein, and a GFF file, which gives sort of location information for all that, all those data, location within the genome. The AMRFinderPlus database, the reference gene catalog curation, that's actually linked to PGAP. So if you submit a genome to NCBI, if you have an assembly, you submit it to NCBI and ask NCBI to annotate it, you'll get these 2, AMRFinderPlus and PGAP, are synced up. If you want, there's also, like I said, point mutation, species-specific point mutations in genes, looking for virulence. One thing I want to point out, you can download, if you are interested in trying the command line version, if you do know a little bit of command line, you have some command line skills. I believe all of these tools can be downloaded from Bioconda. The 3 I've talked about, as well as others, I may be mistaken. Bioconda is a really nice thing if you're getting started in bioinformatics, because I have found that the hardest thing with bioinformatics software in terms of figuring out how to use it is actually installing all the other software you need to install to make that particular tool works. And Bioconda does that for you. So if you're starting out, doing more command line work, I would suggest thinking about learning a bit about Bioconda. It's very straightforward. And we actually, on our website, when we describe our tool, we actually tell people how to install Bioconda and how to get it from Bioconda. Just a little plug, hopefully, if people are thinking long-term about maybe getting into command line things. I'm putting this up here not as a tutorial per se, but just to make the point, and all the command line tools, once you've installed it, it's pretty simple to use. I mean, this is really just one line, one command. This is the simplest version. I just have a nucleotide file and an output file that spits out a table in tab delimited text that you can open in Excel. There are more complicated things, so you can put in a protein set of annotated proteins. Here would be my genome, set of annotated proteins, the file that sort of links up where these proteins are on the genome, an output file. If I want to say, well, I want to run an E. coli specific analysis, I can do that. And then also, do I want to include these genes? So what I want to sort of end with, or get close to ending with, are 2 examples. One is I call the good, and this is where our tool, as well as the others, I double check them. I ran a salmonella isolate, and you get the expected answer. The genotype matches the phenotype. And then I want to go through a bad example of Pseudomonas where that is clearly not the case, and talk a bit about why that might not be the case. And I've put the commands on here in case you wanted to run it, or even if you just wanted to get the files from NCBI and try it with other tools as well. So just to talk about Salmonella enterica, actually, let me step back. I want to jump off this for a second and just show briefly the output. I hope everyone can see this. I'll try to make it a little bigger if you're on a small screen. This is just the kind of output you get from AMRFinderPlus, and you'd actually see it in our web tool, MicroBiggie. I've just opened it in Excel. so that it's easy to read. I know that the bioinformaticians might be cringing a little at me using Excel, but it is actually easy to read it. You know, with a lot of these tools, just like you saw with the ResFinder output, you get a protein identifier. You're told what contig it occurs on, where in the contig. You get the gene symbol, the sequence name, the kind of, for us, it's this, you know, is it sort of this additional stuff or these core AMR genes? Some kind of this basic description. Is it AMR? Is it stress? Class, subclass. So here's an aminoglycoside. Resistance gene that confers resistance to gentamicin. We described the method. So was it an HMM? Here's a case where there's an internal stop in a gene. So we tell you, yes, you found this gene, but there's actually an internal stop there. So it won't be functional. I'm just going to look at the AMR genes just to show you that, in fact, I did not make that example up. These are just the resistance genes that we found in this particular AMRFinderPlus found in this particular salmonella isolate. Just to go back. So this salmonella, I just showed you this, the resistance phenotype. This was an isolate for which we had resistance phenotypes. It's ampicillin resistant, gentamicin resistant, tetracycline. It was tested, I think, against 14 other drugs, and no other resistances. And this makes sense. We had a TEM1 beta lactamase. It's resistant to ampicillin. That's expected. A gentamicin resistance gene, and these common tetracycline resistance genes. And we didn't find any additional resistance elements or genes that, you know, so there was no discordance there. We also found a streptomycin resistance gene, but that wasn't tested. So this is a good example. Like I said, salmonella is easy. Then we get to Pseudomonas aeruginosa, and what's highlighted in the red here are all the things that went wrong. And Finn asked me to do this, but I think this is actually an important point because it was raised in the first talk. So there are things that were successfully predicted. We have the GES beta lactamase, and we see resistance to cephalosporins. Imipenem, we have some point mutations that confer resistance to fluoroquinolones. We have these gentamicin resistance genes, imipenem and piperacillin tazobactam. We know from the literature, you can this GES2 allele confers beta lactamase confers resistance. But then, for instance, we see these three inhibitor beta lactamase inhibitor combinations. These are relatively new, and while there are mechanisms described in the literature, and in fact, AMRFinderPlus does look for these, and I believe the other tools do have some of these as well. But these are really new. There are papers coming out every month. We didn't find any using our tool. I ran RGI and ResFinder on them as well, and didn't see anything that said, that explains it. So these might be novel mechanisms. Alternatively, there may be some expression going on here. It could be a combination of mechanisms that GES, when there's a porin mutation, for instance, you don't get the inhibitor or the antibiotic coming in at high levels, so you might get resistance. It's unclear. And then there is something even more confusing. Tobramycin and amikacin, we do know a lot about their resistance mechanisms, and we didn't find any resistance mechanisms in this genome. These can also be affected again by efflux pumps, so that may be what's happening here. But this is sort of the bad example. So just to conclude, having ended on sort of that down note, I want to point out that for some organisms, much of the Enterobacteriales, at least many drugs, prediction is accurate, and just based on sort of acquired genes and point mutations. Some bug drug combinations are challenging. New phenotypes are often inadequately understood, and there's also other things like porins and various other mechanisms that can be broken or may be altered, and that's a hard problem right now for a lot of these tools to get at. You can describe individual lesions, so here's a stop mutation or here's a change, but what do you do when there's a frame shift 80% of the way in, and we might not even understand the biology to know if that matters or not. In general, when I look at the literature, organisms like Pseudomonas and Acinetobacter are hard, and that's partly due to less characterization and partly due to more complex and more multifactorial mechanisms of resistance. And I talked about this earlier, that you can throw everything at Pseudomonas aeruginosa and sometimes it just doesn't work. So in general, in short, I think you should use the appropriate tool or tools for your needs. The methods do matter. Database quality to considerably matters, although I think the larger tools are borrowing from each other extensively. So I think those gaps have been closed. And you also have to think about what answers are you trying to get? What output do you need to answer the questions you have? Just to end, because I covered a lot here, and this will be available on the slides we send around. There are a bunch of resources. These were talked about earlier on. Our catalog of HMMs, our catalog of genes, the isolate browser, which gives you sort of an isolate level view for over 950,000 isolates. MicroBiggie, which gives you sort of a gene element view for about 770,000 isolates. And there's a whole bunch of search syntax and filters you can use to say, for instance, in MicroBiggie, show me all the things that have KPC2, as well as show me all the contigs that have KPC2. So there's some nice research applications there. Reference gene hierarchy. I talked about that. We actually now have a display. So if you're trying to figure out, well, why did AMRFinderPlus give me that answer? Maybe that will help you. And if you have any questions about this or general issues, you can contact me at pd-help, then at the NCBI website. And just to finally sum up the work that I do, it's part of a much larger group that's involved in both the pathogen detection project, and the people I've bold-faced are involved in various parts of our AMR efforts. And I just want to highlight our NARMS collaborators who've helped us on the sort of making sure that we got the genotypes right by giving us good phenotypes. And again, if you have any questions, feel free to contact us here. And I'll stop there.
Thanks very much, Dr. Feldgarden. That was a really excellent overview of the trials and tribulations of AMR gene detection tools. So there's a few questions, and you touched on quite a few of them, but it might be good just to kind of hit them again, especially in some of them are ripe for later panel discussion. But how do you know when an AMR gene is still an AMR gene? If you've got your reference allele and it's diverged a certain percentage, how do you know it's still an AMR gene or not? Like, as you said, one rule for all versus custom for gene families.
Yeah, so I mean, that's always a problem. And in fact, that's why we have ongoing curation. I don't pretend that we or anyone else gets it right. The HMMs are usually pretty good. And the way I think about it is, an HMM is, again, 100% match, what that is because somebody's gone out and characterized it. It's something that's, a small bit divergent. You probably feel pretty good. In fact, for some things like tetracycline resistance genes, the standard in the field is if it's 80% identical or higher, we call it a tetracycline resistance gene. That might be too broad, but it is sort of, we try to correlate genotype and phenotype at times, but there is sort of I see it going from an exact hit to a close hit to something, let's say in our tool, identified by an HMM. And as you move farther away, you should have less confidence. If I were looking at an HMM hit, let's say in a sort of non-standard pathogen, and somebody said, well, should I treat or not treat based on that? I might be hesitant. But, a high identity blast state, you probably have a pretty good idea what it's going to do. So I know that's not a satisfying answer, but that's the reality of the biology. And sometimes it doesn't matter. Like I said, where you get these tiny little changes that can affect the phenotype, in many cases, they don't really seem to, you can have considerable diversity in the family of enzymes. And either nobody's characterized it or nobody cares, and that seems to be good enough based on sort of when you do these genotype phenotype comparisons. So there isn't really one hard, fast rule. I think it's important to understand the limitations and approaches of your tools and take that into account when you're trying to make that assessment.
Yeah, and much the equivalent of the HMM in AMR Finder Plus is in RGI. The loose category kind of covers a lot of those similar possible ones. What do you see as the kind of, and again, you did touch on this, but the kind of different pros and cons of the HMM versus protein blast versus nucleotide blast in terms of sensitivity and specificity of your hits?
Yeah, so I mean, the problem with HMM, I mean, someday there are now a bunch of these metagenomic samples where people go through and they clone genes out and ask, is this thing a resistance gene or not, right? So I think we can really start to test those. The HMMs, if you have, especially with proteins, if you have an annotation, they can be very fast. So I mean, for instance, when the first MCR, mobile colicitance gene, was discovered, we had about 300,000 isolates in the pathogen detection system at that time. We ran the HMMs, and sure, we did some parallelization and not that, but in under a day, we screened 350,000 genomes for the presence of this gene so we could go through and identify it. So they are fast, and they're typically good for casting a wide net, and they are based on biology. Protein, I'm partial to protein for some sort of technical reasons, in that, at least in our tool, it's often easy to detect frame shifts with protein sequence if you also have a nucleotide sequence, because you can map the two and say, here's this protein, but this thing switched to a stop codon. And protein does give you the function. So for instance, with beta-lactamase alleles, the curatorial standard is based on protein sequence. So you want, for that, you want to start there. Nucleotide approaches, if I think about something like, particularly with metagenomic samples, they can be very fast. And the advantage is you don't have to run an annotation. And for instance, if you want to just start with a bunch of reads off the machine, you don't want to, you would go insane if you tried to annotate reads. So using various nucleotide approaches, Kmer or BLAST, can get you there. So I think those are sort of the differences. For our work, when we're doing surveillance and our tools are used by FDA, by, let's say, the Food and Drug Administration, other government agencies, we really want to be conservative. So we like having the protein and the nucleotide there. So again, it's sort of what your ultimate end use, that will also affect things.
Great. Okay. So another one I'm actually interested in myself is, you mentioned the issues of insertion elements disrupting AMR genes and broken genes. Are there any tools out there that actually identify, are robust to that, or is it better to kind of be a bit more?
I don't want to make a mistake and say there aren't any, because that might reflect my ignorance. There are a lot of tools out there. The sort of big three I talked about, there really aren't, and some of the others, there are. So for instance, and some of them are organism specific. So Collaborate, for instance, with the OmpK porins will actually sort of give you a read on how long the blast hit is. So in other words, that's one way to look at the gene, right? Is it broken? You have a frame shift somewhere. Well, if you only recovered 17% of the gene, it's probably not functional, and that may confer carbapenem resistance. For things like, I know with AMR Finder Plus, we can identify internal stops, as well as we identify if something is over 50% of the length of a reference, we will call that a partial. In fact, we even distinguish between the partial at the end of a condig versus partial in the middle of a condig. That may be indication of interruption. I think these tools can get at it indirectly. With the real thing about insertion sequences that I get concerned about, we're collaborating with a group at Robert Bonomo on some OXA work, is that you have these IS elements that jump into the promoter region. So lots of Acinetobacter have carbapenemases. When the IS element jumps into the promoter region, then the carbapenemase is turned on. And that's something that I haven't seen a whole lot of, but there may be people developing sort of what I would call artisanal tools that detect that. So I think that's part of it. As was talked about in the first talk, we like to develop tools that do it all, but it may be we have to sort of develop species specific, or even just sort of, I don't know what you call them, gene specific, in the case of Acinetobacter, oxygene specific tools that are looking for these insertion elements that pop in here.
So I would say, I think the good recommendation is always speak, if you're working on a particular species or a particular class of AMR gene, always worth speaking to people that also work on that gene and also reading the papers and that look at those kind of questions, because they quite often have sometimes specialist things. So like
That's
TV,
right.
for example, has a whole ecosystem of specialist tools.
That's right.
So, okay, another kind of, I'm trying to group by theme. What about tools for metagenomics and AMR gene detection for metagenomics? What are the kind of trials and tribulations of that? And
yeah, so I mean, again, I think it's there are a couple different approaches people have taken. One is just sort of brute force, and if you have access to a lot of computational power, you treat a metagenome as just a really large genome problem. That can become unwieldy. We haven't done that much of it at NCBI. We are starting to explore using some of the tools we've developed. We have a sort of guided assembler that we've built for our pathogen detection system. And, using that to sort of draw out resistance genes and try to find those quickly in samples and type them accurately. There are approaches like that. And like I said, also the Kmer-based approaches, if you have a library. I think with metagenomics right now, the real challenge is more for time constraints. It's a lot harder to do. If you cast a broad net over a huge data set, you can bog down computationally. So I think the Kmer approaches have been reasonably successful. And there are also, I think, some guided assembly approaches that could work. I'm not as familiar as to, I think certain tools like SRST2, for instance, which is read-based, could be very powerful there, and some of the others. So they're just looking for reads and then trying to figure out what the resistance genes are. But I think, with all those tools, the problem is it's going to be harder to find novel mechanisms just because there's so much computing. I think you sort of have to draw the line a little closer, or you just get bogged down with a whole bunch of false positives.
yeah, as there's a lot of trade-offs when it comes to metagenomics, and I think we might talk about that a bit on the panel discussion. But I think for the sake of that is a rich area of the literature, and there's a lot of active work going on in that area.
Yeah.
I think we will probably, I know there's some questions we didn't get to, but there's always the panel discussion later, so hang around for that. I think we'll move on to the next talk, and thank you very much, Dr. Feldgarden. Excellent.